Hi, this lesson focuses on what happens when the cell cycle and cell division doesn't happen normally or it's abnormal. So we're going to take a look at what checkpoints are, normal versus abnormal cell death, and what can arise if cells do not die when they're supposed to, which is cancer. So in terms of the cell cycle, as each stage or phase progresses along, there are certain checkpoints that produce proteins or molecules that monitor the cell activities in their surroundings. So when the G1 phase is nearly done, certain things that are checked for is cell size. Has it grown enough to progress into synthesis? Are there enough nutrients to allow it to progress? What about growth factors? Is there any damage in the DNA? Now this would be important because the next phase is synthesis, where DNA is being duplicated. So this checkpoint here produces certain proteins that are going to send messages to other areas of the nucleus that are either going to allow it to progress if all of these things are where they should be, or it's going to stop it. So in stopping it, it could make it stay in that phase for longer until the needs are met, or it could take it completely out of the cell cycle and stop. There's another checkpoint at the end of G2, which is again looking for cell size, because this is right before the cell division. So is it large enough? Is there enough cytoplasm to actually split into two? Are there enough organelles? It's also looking to kind of proofread the DNA replication. Did everything happen correctly? Are there the right number of chromosomes? Are they attached correctly? And this is going to make sure that there aren't problems arising during mitosis. In mitosis, there is one checkpoint as well, and this is when everything's checking for spindle attachment. So in mitosis, all those chromosomes line up at the center of the cell. In order to progress into anaphase, making sure that those spindles are attached so that those chromosomes will pull apart properly. If a checkpoint is not met, the cells will remain in interphase. If there's a signal from another cell, so maybe surrounding cells can send some signals out and saying, nope, something's wrong in the environment, or there aren't enough nutrients, but advising that cell to stay in interphase. As mentioned before, if there aren't enough nutrients, nutrients are needed for energy. This is a process that requires a lot of energy, cell division does. So if there aren't enough nutrients, there won't be enough energy to progress. If DNA is not replicated, so if it's not replicated at all, if it's replicated incorrectly, the cell will remain in interphase. If there's any damage in the DNA, that's going to cause those cells to stay in interphase as well. As mentioned in an earlier lesson, there is another phase called this G0 or G0 phase. And this is also called the resting phase. So when cells go into this phase, it can be temporary. So muscle cells will go in it sometimes if they need to kind of stay there, or if there's no need for cell division or repair, they can stay there, or it can be permanent. So in terms of nerve cells, once your nerves are fully grown and your brain is fully developed, they enter a permanent stage of G0. And that's one of the reasons why brain injuries and spinal injuries can be so devastating, is that they are in this rest phase. They don't go through cell division, therefore they're not going to replace those cells or fix any damage. DNA damage will also cause cells to be pulled out of the cell cycle and stay in this G0 phase. Okay, so as mentioned, nerves in the brain is a common area of G0 phase. Now, cell death. Cells are supposed to die. They don't live forever. There are ways that there's controlled death, but then there's also unexpected or uncontrolled death. So necrosis and apoptosis are our two forms of cell death. Now, if we take a look, we have a normal cell. If a cell is going through apoptosis, this is normal. So either the cell, it is old, it is damaged, it is ready to be broken down. It breaks itself down and it is broken apart or phagocytosed. And there is no inflammation or no damage to the surrounding tissues. However, if that cell, say, becomes infected or is not following kind of normal cell death, that cell actually swells up and causes inflammation as it's going through its death. So apoptosis is the controlled death of old cells. And what happens is white blood cells come along and they actually literally take those other parts that are no longer needed anymore, and they are reused within the cells. Necrosis, on the other hand, can be due to external factors. So if somebody is exposed to a toxin, if there's an infection, this is necrosis. So when you think about infections, you think about an area that is warm, that is swollen, and that is because of this swelling due to necrosis. So that also leads us to cancer, because cancer is when cells are not going through cell division normally. So abnormal cell division. Now that can be caused for many things, mutations, carcinogens. We'll get back to that a little bit later. But normal cell division, you have a healthy cell, you have daughter cells, which will then grow, make more daughter cells. If a cell is injured, it will go through apoptosis so that it cannot make other cells that are also injured or damaged. Whereas in cancer cell division, you get that damaged cell and it will continue to go through cell division. It won't be killed off normally. Therefore, all of its daughter cells will have the exact same damage that it had. So instead of apoptosis, the cells divide uncontrollably when in cancer. So basically saying that they don't stay in interphase. They just keep going through mitosis. They rush through interphase, don't stop at the checkpoints, and go back to the cell division. So a couple things when we take a look at this rushing through cell division is those cells are going to build up on top of each other and they're going to pile up and crowd each other, and that is a tumor. So tumors, anytime you have these cells building up on top of one another that don't have an obvious function. Normally when we're having cell division, there's a reason why they are dividing. Is there damage? Is there a need for reproduction? Or why are the cells dividing? In case of a tumor, they're dividing, but there's no reason why. Okay. The other thing about tumors is they can reduce the effectiveness of that surrounding tissue. These cells are trying to continue to do their function, while these other cells are crowding them out, they're pushing on them, they're squeezing on them, and they're also stealing their nutrients. Because mitosis is a high energy process, they're going to steal the nutrients that these other cells need. So they do start affecting the cells around them. So in terms of tumors, we kind of have three different classifications of tumors. Benign, meaning that you have uncontrolled cell division, but those cells are not affecting the cells around them. Yeah, they're crowding them a little bit, they're squishing a little bit, but they are not damaging those cells around them. Whereas when you have a malignant tumor, they are actually directly interfering with those surrounding cells. So they are damaging them. And this is when we call a tumor cancerous, is when it is malignant. Metastasis or metastasizing is when a piece of a malignant tumor actually breaks off and it travels somewhere else and it starts a secondary tumor. So it can get into the bloodstream and travel to any other cell or location in the body, and it starts to interfere with cells there. So if we want to take a look at what that looks like, when you have normal cells, there is a definitive shape and size. Well, the first kind of stage in developing cancer is hyperplasia. It's not cancer yet, but hyper meaning excessive. So if you look at it, those cells are starting to grow and they're starting to crowd each other, but they're still where they're supposed to be and they're still able to carry out their normal function. Mild dysplasia is that next step, is when you start seeing those cells crowding up on top of each other and actually starting to slightly move out of location and pile up on top of one another. Okay, severe dysplasia is now you have severe cell movement. They are getting out of place. That's what dysplasia means, is that those cells are no longer where they're supposed to be. And the final step is actual cancer. And it is deemed cancer when those cells become invasive to other cells around them. Okay, the other thing to remember about cancer is these cells, as they are developing and growing and interfering with the other cells, they can actually stimulate cells beside them to start dividing uncontrollably as well. And that's why sometimes people can start off with one type of cancer and develop another, is those cells actually signal to each other, and basically cancer cells can recruit other cells to become cancer cells as well. So if we want to take a look at what healthy cells look like versus cancerous skin cells, if we zoom in to this one here, you look at it and you see that there is a pattern. Everything is kind of structured the same. The dark spots represent the nucleus, and there's more cytoplasm than there is nucleus. Whereas when you look at a sample of a cancerous skin cell, look at the size of the nuclei. They're larger. There's not as much cytoplasm around them because they're dividing really quickly, means that those cells don't have enough time to grow. There's not enough cytoplasm for them to make two normal-sized daughter cells. Therefore, the nucleus is a larger proportion of all the cells. You start looking at those boundaries. They're all starting to kind of get jumbled together, and there isn't the nice, neat organization any longer. The other thing looking for cancer cells is there will be more cells in the phases of mitosis. Remember, in a normal healthy cell, interphase accounts for 90% of the time. So 90% of cells should be in interphase. Whereas when you start looking at cancer cells, you notice a lot of those cells are in prophase, metaphase, anaphase, or telophase. They're not in that resting or growing stage. So when we actually look at appearance of cancer cells, normal versus cancer, you take a look and you say, okay, are they large? Are they irregularly shaped? How big is the nucleus compared to the rest of the cell? How much cytoplasmic volume is there? Do they have regular shape or do they vary? Specialized features, right? We talk, each cell shape dictates function. So is it starting to lose its typical shape that allows it to have its function? Is there evidence of disorganization? And are there poorly defined boundaries? In a tumor, it can be a benign tumor, which is not cancerous. You can clearly see where those cells, where their boundary or their edge is. Whereas a poorly defined tumor boundary will most likely indicate that it's probably a malignant tumor.